day to Dr. Sharmila. We are from Group 2 and we need to design an experiment in JSP for the impact of pH and temperature on enzyme activity in cell-free systems. I am Kalshini Manimaran and my matrix number is 225544. First, we identify the experimental unit and variables. The experimental unit is the entity or item that receives a treatment in an experiment. Experimental units must be independent of one another to avoid bias in results. This means that the treatment applied to one unit should not influence the outcome of another unit. In this experiment, a sample of the cell-free enzyme preparation is the experimental unit. For example, we conduct an experiment to test the effect of pH and temperature on amylase extraction. Hence, the experimental unit is the amylase extraction. Next would be the factor of this experiment. Factor is basically an independent variable controlled by the experimenter, which in a simple word is that the variable we manipulate to observe its effect on the response variable. In this case, the factors are the pH and temperature. Both of them are numerical factors. Just now we know there is two factors in this experiment. Now we identify the level of this experimental design. Level is the different values of a factor. So for this experiment, the level for the factor pH could be pH 4, pH 6 and pH 8, while the factor temperature could be 25 degrees Celsius, 30 degrees Celsius and 35 degrees Celsius. Each factor can have multiple levels which represent the different conditions under which the experiment is conducted. Next, we dip into the response variable. A response variable, also known as a dependent variable, which is the variable in an experiment or study that we measure or observe to assess the effect of factors in the sample. It is the outcome or result that are we interested to analyze and understand how it is influenced by the changes in the independent variables, which is the factors, pH and temperature. In this experiment, the response variable is the enzyme activity, which we measure in different levels of factor.
Now, we'll move on to completely randomized design, CRD, which is a fundamental experimental design that is widely used in statistics to evaluate the effects of single factor on a responsible variable. To explain the principles of CRD in our context of data, we will analyze it according to four criteria, which are different levels, controls, replicates, and randomization. Firstly, in our experimental design, we have two factors, temperature in degrees Celsius and pH. Each factor has multiple levels that we tested across different experimental runs. For instance, temperature levels include 25, 30, 35, 37, and 45 degrees Celsius, while pH levels are set to 4, 5, 6, 7, and 8. This setup allows us to observe how variations in both temperature and pH can affect the response variable. By including multiple levels for each factor, we can effectively assess their individual and interactive effects on the outcome. Next up, we'll look at control. Control in an experimental context refers to a standard or baseline condition against which experimental treatments can be compared. It serves as a reference point to help determine the effects of variable being tested. In our context of our CRD experiment, the control treatment would typically involve maintaining conditions that are known to support optimal enzyme activity. For instance, using a pH of 7 and temperature of 37 degrees Celsius as a control treatment allows us to measure enzyme activity under neutral pH and physiological temperature conditions. This setup provides a reliable baseline for evaluating how deviations from these conditions, such as lower or higher pH levels or different temperature, can affect the enzyme performance. Now, I'll pass on this part to my friend Afdal.
Moreover, replication is vital for enhancing the reliability of our results. In our factorial design, we made sure that each level of temperature and pH is replicated multiple times to account for natural variability in the data. For instance, we have two replicates for each temperature and pH levels, which allows us to obtain a more accurate estimate of the treatment effects and improve the statistical power of our analysis. And this replication can be seen in the total number of runs. Lastly, randomization is a core principle of CRD that helps eliminate bias in treatment assignment. In our factorial design data, we use a randomization method that uses the RAND function. And that is done to assign different treatment combinations to experimental runs. This random assignment ensures that each treatment has an equal chance of being tested in any given run order. By randomizing the order of runs, we mitigate potential confounding of variables that could influence our results. And by doing that, we increases the validity of our conclusion.
To construct a completely randomized design, the first step is to identify the level involved for each factor. The level involved for the factor pH are pH 4, 5, 6, 7, and 8. And each level consists of 2 replicates. For the factor temperature, the level involved are 25 degrees Celsius, 30 degrees Celsius, 35 degrees Celsius, 37 degrees Celsius, and 45 degrees Celsius. And each level also consists of 2 replicates. The control for each factor is include, which are 7, pH 7, and 37 degrees Celsius. Hence, there is a total of 10 runs for each factors. Next, randomization is the key. The random function is used to generate random value between 0 and 1 for each run. Once random numbers are generated, sort the row in ascending or descending order based on the value to randomize the experimental run. This approach maintains complete randomization, which is critical for the integrity of the experiment, additionally ensuring sufficient replication of treatment and validating the randomization process enhance the reliability of result. CRD conduct this way is straightforward and minimizing bias, making it a robust choice for many experiment setup. In short, by employing a completely randomized design and utilizing the provided table for treatment assignment, we can effectively analyze how different pH level and temperature impact enzyme activity in a controlled manner. The randomization process enhances the validity of our result, making it easier to draw meaningful conclusions from our data analyzed using ANOVA.
Next, we'll now explain the two-level factorial design. It is a tool to experiment on many factors simultaneously. The simplest factorial design involves 2 factors, each at 2 levels. It contrasts averages, thus providing statistical power to the effect of estimates. We'll now illustrate the basic principles of the two-level factorial design through an example, which is the impact of pH and temperature on enzyme activity in the cell-free system. In this example, 2 factors are studied, which are the pH and temperature. Each factor has a low and a high level. While deciding on the levels can be set as widely apart as possible to increase the likelihood of an effect, but we have to stay within the operating boundaries. This means that the low and the high levels of the pH and temperature should not be too high or too low until the enzyme becomes inactive or denatured. Hence, the low level of the pH set is pH 4 and the high level is pH 8, whereby the low level for the temperature is 25 degrees Celsius and the high level of the temperature would be 45 degrees Celsius.
In this experiment, 2 factors are included, so there will be 4 unique combinations with 2 center points. Hence, the number of runs will be 6 runs. The order of experimental run is randomized to satisfy the statistical requirement of independence of observation and minimize the risk that external factors influence the result. In the two-level factorial design, the experimental run is randomized using the rand function to generate and sort the random numbers. In addition, replication can be conducted by conducting multiple experimental runs with the same factor setting. For example, there are 6 runs for 1 replicate in this experiment. If 2 replicates are conducted, another 6 runs with the same setting will be repeated. Conducting replication improves accuracy by increasing the number of data points available for analysis to detect random errors and provide a more reliable result. Moreover, a control treatment can also be prepared to serve as the reference point to compare the experimental outcome. The optimum pH 7, and temperature 37 degrees Celsius will be the control of this experiment to compare the enzyme activity at low and high level temperature.
Furthermore, replicated center points are included in the two-level factorial design to detect non-linearity. This is important because factorial designs assume there's a linear relationship between the factors and the response. If there is non-linearity, the results might be misleading. Hence, the replicated center points are crucial for the curvature test.
So now I will compare between completely randomized design and two-level factorial design. First, completely randomized design is ideal for identifying treatment effect in a simple setup, especially when there is no environmental variations and all the experimental units are similar. In contrast, two-level factorial designs is better for studying the main effect and interaction between factors, offering an initial understanding of the relationships. While completely randomized design focuses on one factor with multiple levels, two-level factorial designs involve two or more factors, each with two levels, such as height and low. Therefore, completely randomized design is suitable for control and homogeneous conditions, whereas two-level factorial designs is more effective in preliminary testing between multiple factors.
Now I will be explaining about advantages and disadvantages of completely randomized design (CRD) and two-level factorial design. CRD's advantages for its simplicity and ease of implementation, making it popular choice for experiments with homogeneous conditions. Its flexibility allows it to adapt to various types of experiments without requiring complex grouping or blocking. However, CRD has significant limitations, such as its inability to control variability effectively in heterogeneous environments, leading to less reliable results when experimental units differ. Additionally, CRD is limited in its scope as it does not account for interactions between factors, making it unsuitable for multifactorial studies. In contrast, a two-level factorial design excels in studying multiple factors simultaneously, efficiently exploring both main effects and interactions while minimizing the number of experimental runs compared to testing each factor individually. This makes it particularly valuable for understanding complex systems. However, its use of only two levels per factor can oversimplify real-world scenarios, especially if non-linear relationships exist. Furthermore, the designs become more increasingly complex and resource-intensive as the number of factors grows, making it challenging to manage large-scale experiments.